Class 12. Today we will start the second chapter of your syllabus, Sexual Reproduction in Flowering Plants. I have divided the chapter into 3 video clippings. This is the first video clipping. Here we will discuss about microsporogenesis, megasporogenesis and double fertilization. Microsporogenesis is the process of formation of microspores or pollen grains. Megasporogenesis is the process of formation of the functional megaspore, and double fertilization involves 2 events of fusion between the male gamete with the egg cell and with the secondary nucleus. Let's start with microsporogenesis. This is a transverse section of an anther. Anther, as you know, is a part of the androecium of a flower. The circular colored structures that you find here are the chambers of an anther. One such chamber or microsporangium has been enlarged here, where you can see these blue dots that I have drawn here. You can see the blue dots in a better view here. These blue dots represent the pollen mother cell, abbreviated as PMC. These are the diploid cells. One such pollen mother cell undergoes the first meiotic division to give us 2 haploid cells. They further undergo the second meiotic division, giving 4 haploid microspore cells. Together they are called microspore tetrad. They will later develop into the male gametophyte. This blue colored line that you see here, the pollen tube, the male gametophyte, which contains the tube nucleus and the generative nucleus. Now let's come to megasporogenesis. We can see here the internal structure of an ovule. This pink dot that you find here is the megaspore mother cell, abbreviated as MMC. This is also again a diploid cell. It undergoes the first meiotic division, giving 2 haploid megaspores, which further undergo the second meiotic division, 4 haploid megaspores. Here we find something different occurring. Out of these 4 haploid megaspores, 3 will degenerate and only 1 will persist as functional megaspore, which will later develop into the female gametophyte. Next we come to double fertilization. As you know, this is the pistil of a flower showing the stigma, style and ovary. Numerous pollen grains may come and fall on the stigma, but one will develop as the pollen tube, this blue line, where you can see 2 white dots. They represent the male gametes or the sperms. Now the pollen tube contains tube nucleus and generative nucleus, out of which the tube nucleus degenerates and the generative nucleus gives us the 2 male gametes or 2 sperms. And within the female gametophyte, we find the 3 antipodal cells, polar nuclei or the secondary nucleus, which is diploid, egg cell, which is haploid, and on either side of the egg cells, we find 2 synergids. Now, the first fusion occurs between one male gamete or sperm and the egg cell. So, one male gamete or sperm haploid plus egg cell, again haploid, giving us a diploid zygote, which will later on develop into the fruit. This process is called syngamy. And here, the second male gamete or the sperm will fuse with the diploid polar nuclei. So, another male gamete or sperm haploid plus polar nuclei, which is diploid, will give rise to a triploid structure called endosperm. This process is called triple fusion. So, since there are 2 events of fusion, syngamy and triple fusion, together we call it double fertilization. And at the end of fertilization, the 2 most important structures that are formed are the embryo and the endosperm. Now, in your textbook, you will find the diagram of the stages of monocot embryo and dicot embryo development. So, you need to practice those stages. And endosperm, according to the process of formation, endosperm may be of 3 types. Nuclear endosperm, where the primary endosperm nucleus divides repeatedly without wall formation to produce a large number of free nuclei. Second, cellular endosperm, where we find the primary endosperm nucleus is followed by cytokinesis and cell walls are laid successively. And the third type is halobial endosperm. This is an intermediate type between cellular and nuclear endosperm. So here we come to the end of these three processes: microsporogenesis, megasporogenesis, and double fertilization. Thank you.